metabolism, and we're going to go ahead and work through this on a video recording, so I hope you get an opportunity to watch this, and if there's sections on it that you want to go back and take a look at again, you certainly can do so. So this will be posted, and there should be a link on Moodle and available through YouTube. Okay, so here we go. Let's see here. Okay, the energy of life. So the living cell is a miniature chemical factory where thousands of reactions occur. What we're going to focus on in this chapter is cellular respiration, which takes energy stored in sugars and other fuels, and it will apply this energy to perform work. Some organisms even convert energy to light, as in bioluminescence. Okay, metabolism, some definitions. Metabolism is the totality of an organism's chemical reactions. Metabolism is an emergent property of life that arises from orderly interactions between molecules. A metabolic pathway begins with a specific molecule and ends with a product. Each step is catalyzed by a specific enzyme. As shown in this diagram here, you can see going from A to D, you go through at least 3 reactions. You start with molecule A, and each enzyme along the way facilitates each step, proceeding on towards the direction of D. Now, catabolic pathways release energy by breaking down complex molecules into simpler compounds. Cellular respiration, the breakdown of glucose in the presence of oxygen, is an example of a catabolic pathway. Anabolic pathways consume energy to build complex molecules from simpler ones. For example, the synthesis of protein from amino acid is considered an anabolic pathway. Bioenergetics is the study of how energy flows through living organisms. Now, energy is the capacity to cause change, and energy exists in a variety of forms, some of which can perform work. Here are some of the major types of energy that we see in biology. Kinetic energy, the energy that's associated with motion. The easy way to remember that is if you're familiar with like kinesiology or kinetics, movement. So you can think of the energy of motion. Thermal energy, so therm or thermos, like heat. So thermal energy is the kinetic energy associated with random movement of atoms or molecules, and heat's the thermal energy and transfer between objects. Potential energy is energy that matter possesses because of its location or structure. It has the potential of doing something. And then chemical energy is the potential energy available for release as a result of chemical reaction. Energy can be converted from one form to another. Here you can see you got a diver on the platform above the water. He's got a lot of potential energy, more than anybody, just because of his location in relation to the surface of the earth. Then you have to the right, going clockwise, a person diving into the water. They're converting some of their potential energy, or a lot of their potential energy, into kinetic energy. Then you can see that person that, if you will, previously dove into the water is now in the water in the bottom right corner, and now they have less potential than the person on the platform. And then the person to the bottom left that's climbing back up the ladder is converting kinetic energy back into potential energy by lifting their height above the surface of the earth. Okay, thermodynamics, the study of energy transformations. So dynamics is always referring to change, and thermo, heat, so energy transformations. And isolated systems, such as that approximated by a liquid in a thermos, is unable to exchange energy or matter with its surroundings. In an open system, energy and matter can be transferred between the system and its surroundings. Organisms are open systems. Now, according to the first law of thermodynamics, the energy of the universe is constant. It can be transferred and transformed, but it cannot be created or destroyed. The first law is also called the principle of conservation of energy. So here you can see first law of thermodynamics on the left, where you have a chemical energy in food being converted, and then on the right, second law of thermodynamics, the kinetic energy is being released as heat, and carbon dioxide and water molecules as they expel that the gas and moisture from the lungs. Okay, during every energy transfer or transformation, some energy is unusable, and we lose that as heat. That was that second law of thermodynamics we saw on the previous slide with the bear running through the water. Every energy transfer or transformation increases the entropy of the universe, and entropy is a measure of molecular disorder or randomness. Living cells unavoidably convert organized forms of energy to heat, which is a more disordered form of energy. Spontaneous processes occur without energy input. They can happen very quickly or very slowly. For a process to occur spontaneously, it must increase the entropy of the universe. Processes that decrease entropy are non-spontaneous. They will occur only if energy is provided. So don't get spontaneous confused with instantaneous. A lot of people tend to do that. Spontaneous means that it will go at some point in the future without adding any kind of energy to it. It may take a million years, but the reaction will actually go without adding any outside energy to it. Something that's instantaneous means that's time dependent, right? We're going to consider that relationship according to how quickly that occurs. Spontaneous means it'll just happen at some point in time. Okay, organisms create ordered structures from less organized forms of energy and matter. Organisms also replace ordered forms of matter and energy in their surroundings with less ordered forms. For example, animals consume complex molecules in their food and release smaller, lower energy molecules and heat into the surroundings. So here's a picture of kind of some different relationships of order, but you can see there's certainly order in these organisms. The evolution of more complex organisms does not violate the second law of thermodynamics. Entropy, or disorder, may decrease in a particular system, such as an organism, as long as the total entropy of the system and surroundings increases. So biologists want to know which reactions occur spontaneously and which require input of energy. To do so, they need to determine the energy and entropy changes that occur in chemical reactions. A living system's free energy is energy that can do work when temperature and pressure are uniform, as in a living cell. So this is called free energy change, which we always represent change with delta. So this is delta G for Gibbs free energy. Okay, the change in free energy, delta G, during a process is related to the change in enthalpy, which is the change in total energy, or delta H, and the change in entropy, which is delta S. And temperature has to be in Kelvin, so make sure if you ever do a problem like this that you do convert it to Kelvin units. So delta G is equal to the delta H minus the temperature times delta S. Okay, so delta G is equal to the change in enthalpy minus temperature times the change in entropy. Okay, delta G is negative for all spontaneous processes. That's really important to remember. So a lot of times you'll see these problems, it'll have delta G and that the value is negative. So if delta G equals something, but that something is negative, then you know that it's going to be a spontaneous process. If it's 0 or positive, it's not going to be spontaneous. Spontaneous processes can be harnessed to perform work. So free energy is a measure of a system's instability, its tendency to change to a more stable state. And during a spontaneous change, free energy decreases and the stability of a system increases. Equilibrium is a state of maximum stability. A process is spontaneous and can perform work only when it is moving towards equilibrium. Okay, so at the top here you got some different diagrams here. You can see a diver, you can see molecules separating, and then you can see molecules breaking apart as in a chemical reaction. So you have gravitational motion in A, diffusion in B, and chemical reaction in C. So what it says here on the left, you have more free energy, higher G means you're going to be less stable with greater work capacity. In a spontaneous change, the free energy of the system decreases. Delta G will be less than 0 as the system becomes more stable, and the released free energy can be harnessed to do work. This will lead to less free energy, or a lower G, but more stability and less work capacity. So the concept of free energy can be applied to the chemistry of life's processes. In an exergonic reaction, you may remember X being going out and En going in, but an exergonic reaction proceeds with a net release of free energy and is spontaneous. So it's spontaneous again, and free energy is going to be released. Endergonic reaction means you're going to absorb free energy from the surrounding environments, and it's not going to be spontaneous. So if you look here at the top, you have an exergonic reaction. It shows that energy is being released, and it is spontaneous, meaning that delta G is going to be less than 0 or negative. In the bottom picture, you have endergonic reaction. You're going to require energy to be put in. It's non-spontaneous because you have to put energy in, and it's going to equal 0 or be greater than 0. Generally, it's going to be greater than 0. Okay, reactions in a closed system eventually reach equilibrium and then can do no work. So here you get delta G, which is less than 0, meaning it's spontaneous, so you have work being performed and exemplified by the light bulb being lit up. On the right side, you have delta G equal to 0. You have stability there. So you don't have any work being performed, right? You've got equal amount of water on both sides, so you don't have a water flow causing the turbine to perform work and generate electricity. Cells are not in equilibrium. They are open systems experiencing a constant flow of materials. A defining feature of life is that metabolism is never at equilibrium. A catabolic pathway in a cell releases free energy in a series of reactions. So here you have an open hydroelectric system. At the top you have delta G less than 0, so you have work being performed, the lights on because the turbine's rotating, causing the production of electricity. And here you can see a multi-step hydroelectric system in the lower picture, where you can see one system's leading is out of equilibrium, so it's causing another system to be out of equilibrium and another system to be out of equilibrium, in which case they're all performing work. Okay, speaking of work, we're going to talk about a little bit about ATP, and just going back real quick to Gibbs free energy and enthalpy and entropy and stuff. There's a lot of videos on YouTube and on the internet that you can take a look at with regards to this. If you still have, I know they can be somewhat of a complex, vague topic, but if you need a little bit of a better explanation, you might be able to find some videos there that use different examples that may click with you or make more sense if you're kind of struggling with that. I understand that process can be kind of a little bit challenging, but please pay attention to that because it is very important for cellular mechanisms. Okay, so ATP. A cell does 3 main kinds of work: chemical, transport, mechanical. Chemical work means you're pushing endergonic reactions. Transport work, you're pumping substances against the direction of spontaneous movement. And mechanical work, such as contraction of muscle cells. To do work, cells manage energy resources by energy coupling, the use of an exergonic process to drive an endergonic one. Most energy coupling in cells is mediated by ATP. Let's see here. ATP, which is adenosine triphosphate, is the cell's energy shuttle. ATP is composed of ribose, a sugar, adenine, a nitrogenous base, and 3 phosphate groups. So it's generally depicted as you see in A, the structure of ATP up at the top. You get the 3 phosphate groups, which are PO4 3 negative, I believe, connected to a ribose sugar, which is a pentose type sugar, and adenine, your nitrogenous base. In the lower picture here, we're showing the hydrolysis of ATP. If you remember, hydrolysis is the breaking apart of something by adding water to it. In this case, you add water to ATP in certain conditions, and you can release that phosphate and some energy. One of that third phosphate group will break off, releasing energy, and then you're left with ADP and an inorganic phosphate and some energy released. The bonds between the phosphate groups of ATP's tail can be broken down by hydrolysis. Energy is released from ATP when the terminal phosphate bond is broken, and this release of energy comes from the chemical change to a state of lower free energy, not from the phosphate bonds themselves. Okay, here's just another picture of it again, just to refresh. And again, that last statement here, one more time, is really important. The release of energy comes from the chemical change to a state of lower free energy, not from the bonds themselves. We tend to think from chemistry that it's always the release of energy due to the breaking of the bond. Not exactly the case. So the 3 types of cellular work, mechanical, transport, and chemical, are powered by the hydrolysis of ATP. In the cell, the energy from the exergonic reaction of ATP hydrolysis can be used to drive an endergonic reaction, and overall the coupled reactions are exergonic. So ATP drives endergonic reactions by phosphorylation, which is transferring a phosphate group to some other molecule, such as a reactant. The recipient is now called a phosphorylated intermediate. So here you can see, we're going to take a look at this reaction. The overall reaction at the top in A is glutamic acid plus ammonia, and it's going to produce glutamine. and it's going to be a delta G for glutamine or glutamic acid of plus 3.4 kilocalories per mole. So that's glutamine. Converting it with ATP hydrolysis, you're going to have, if you break down this process a little bit more closely, you're going to have glutamic acid is going to react with ATP, and then you're going to get this phosphorylated intermediate, which is going to be your glutamic acid with a phosphate bonded to it in the middle, and an ADP now, because you broke that phosphate off the ATP and added it to the glutamic acid, so now you're down to ADP, two phosphates attached to the adenosine diphosphate, and then you're going to add that ammonia in that second step, okay, and now that's going to form glutamine plus ADP and an inorganic phosphate. So that's a little bit more of a breakdown of what's actually happening in A and step B. And then finally, let's take a look at the overall energy. So if you take a look at this, you have glutamic acid on the left plus ammonia plus ATP. Now if you look at glutamic acid and ammonia, when you put those together forming glutamine, you have a plus 3.4 kilocal per mole. Okay, so that's positive, so that's not spontaneous. Okay, but ATP, the delta G of ATP is negative 7.3 kilocalories per mole, right? And that's going to break down into ADP and the inorganic phosphate. So the breakdown of ADP and ATP into ADP plus P is going to be negative 7.3 kilocalories per mole, added to the delta G for the glutamine, which is plus 3.4 kilocalories per mole, and you have a negative net delta G of negative 3.9 kilocalories per mole, which means it's negative, which means it's spontaneous, which means the reaction will go because you've added ATP to it. Without the ATP, they just sit and kind of look at each other indefinitely. Okay, so transport and mechanical work in the cell are powered by ATP hydrolysis. ATP hydrolysis leads to a change in protein shape. In other words, its three-dimensional conformational shape will change, or its shape will change, and it will also, as a result, change its ability to bind to different things due to the relocation of positives and negative charges. So its electronegativity potential in different areas of that new molecule will change shape in that protein. Okay, so transport work, mechanical work. Take a look at a couple of these here. In A, you have transport work, where you add ATP in to a transport protein. It's going to cause it to change, or you get a conformational change, which will allow the solute to move from inside the membrane to the outside of the membrane across the lipid bilayer. Okay, and then the ATP will be broken down to a phosphate and ADP, which will then get released as it changes shape. So that's how a transport protein works across the membrane. In the lower picture, mechanical work, you've got ATP, which is going to stimulate the legs on the motor protein, if you will, the legs which are carrying a vesicle along some kind of filament inside the cell, microtubule or microfilament, and as a result, the ATP binding to the leg, which will break down to ADP, and the inorganic phosphate will give it some energy, which will cause it to break away and release. So it looks like this molecule, this motor protein, is actually walking along this microfilament or microtubule, but in effect, it's actually being stimulated to detach or change shape, detach, reattach a little bit further onto the microtubule and microfilament. So it looks like it's almost walking, because an ATP will hit one leg and then the ATP will hit the opposite leg and then back and forth. So it looks like it's actually kind of waddling along the internal cytoskeleton of the cell. Okay, regeneration of ATP. ATP is a renewable resource that is regenerated by the addition of a phosphate group to adenosine diphosphate. So you can turn it from ADP back into ATP. The energy to phosphorylate ADP comes from catabolic reactions in the cell. The ATP cycle is a revolving door through which energy passes during its transfer from catabolic to anabolic pathways. Okay, here you can see you have energy from catabolism to exergonic, energy releasing processes. So you have ATP plus water, so it's going to undergo hydrolysis, it's going to break down into ADP plus P, which will release energy that will provide cellular work, endergonic energy consuming processes, and then the ADP and inorganic phosphate will reform back into ATP, which will which will again recycle and allow you to go through this process again and again, around and around and around. Now, enzymes speed up metabolic reactions by lowering energy barriers. So, catalysis is a chemical agent that speeds up a reaction without being consumed by the reaction. That's important. It doesn't get utilized in the process. An enzyme is a catalytic protein. For example, sucrase is an enzyme that catalyzes the hydrolysis of sucrose, the sugar. So, ACEs are enzymes, OSes are your sugars. So here you got sucrose on the left, C12H22O11. You can tell it's a carbohydrate because it's got the basic H2O formula. You've got 2 hydrogens to every 1 oxygen, so 22 hydrogens to 11 oxygens. So you got H2O really there. It's like water molecules attached to carbons, 12 carbons. So it's your basic CH2O. So carbohydrate, sucrose plus water. Undergoing hydrolysis in the presence of sucrase is going to break down that sucrose into glucose and fructose. If you notice, glucose and fructose have the same chemical formula, but they are obviously different in their structure. So you get on glucose, you have the hydroxyl unit that's added in, and the hydrogen from the other water molecule that's used in hydrolysis gets attached to the free oxygen on the pentose fructose sugar. So the H attaches to the O, and the water molecule gets used up, and you have now 2 independent molecules. If you notice, you have 12 oxygens attached on the right side and 24 hydrogens if you add the H's and O's from the glucose and fructose together, whereas you only have the 22 and 11 hydrogens and oxygens respectively in sucrose, so that water gives them the extra 2 hydrogens and 1 oxygen to be able to become 24 H's and 12 O's. Okay, the activation energy barrier. Every chemical reaction between molecules involves bond breaking and bond forming. The initial energy needed to start a chemical reaction is called the free energy of activation, or the activation energy, E sub A. Activation energy is often supplied in the form of thermal energy that the reactant molecules absorb from their surroundings. In other words, some sort of heat of some sort. Okay, so here on the left side you've got the reactants, the energy of activation is applied, perhaps through heat of some sort. The molecules go through a transition state where bonds get weakened, and eventually they break and reform, and now A bonds to C and B bonds to D. You get a delta G less than 0, so it's a spontaneous reaction, and this thing goes forward. In catalysis, enzymes or other catalysts speed up specific reactions by lowering the activation barrier. Enzymes do not affect the change in the amount of free energy. Instead, they hasten reactions that would occur eventually. So that's one of the things that enzymes make reactions go, but a lot of times I think we're under the impression that they actually do something energetically favorable to the chemical reaction and cause it to speed up the process, like they're adding energy or something to that. And a lot of times it's not the case. They're just going to speed up the process in which the reaction would actually occur. Remember, it's a spontaneous reaction anyways. They're just speeding up the process. So here you go. If you take a look at this diagram on the left, the y-axis you have free energy, and the x-axis you have the progress of the reaction. And you can see here, if you look at your standard, which is the black line, the course of the reaction without the enzyme, that's normally how it would go. Okay, and it's showing you the energy of activation without the enzymes, the vertical black line, which is your differential between the on the x-axis there, between reactants and products being formed or breaking down. So the energy of activation without the enzyme is shown to be much higher than it is with the energy of activation enzyme. When that's added, it'll be lowered significantly, demonstrated by the red arrow. And so you can see the delta G is really unaffected by the enzyme. The amount of energy that's put into the system is not necessarily there. What changes is the reaction time. So the energy of activation barrier gets lowered. So the reactant that an enzyme acts on is called the enzyme substrate. The enzyme binds to its substrate. It forms what's called an enzyme substrate complex, and while bound, the activity of the enzyme converts substrate to product. The reaction catalyzed by each enzyme is very specific. There's an active site is the region on the enzyme where the substrate will bind. Okay, that's important to always pay attention to where these are. Induced fit of a substrate brings chemical groups of the active site into positions that enhance their ability to catalyze the reaction. So here you can see on the left you have an enzyme. It's got an active binding site on the inside. You have a substrate. And then when the substrate binds into that active site, it causes a change called the enzyme substrate complex. You can see that it kind of maneuvers around a little bit and changes. And if you look up in this region here, this is a little bit more visible. You can see that this shape up here is slightly different than this here. This curvature here is slightly different than this. So you get a different shaped enzyme as a result of these two bonding. Now this shape may actually bind to another site on in a cell, stimulating another reaction to go, whereas this one wouldn't. Again, it's got an active site here, but it may cause some kind of tertiary bonding as well. In an enzymatic reaction, the substrate binds to the active site of the enzyme. Enzymes are extremely fast acting and emerge from reactions in their original form. And very small amounts of enzyme can have a huge metabolic effect because they're used repeatedly in catalytic cycles. Remember, they can be recycled. So here you have substrates that enter into an active site, forming an enzyme substrate complex. Okay, the active site will then lower the activation energy, hastening it. The substrates are converted into new products through the change in that shape. The products are released, and then the active site is available for substrates to rebind to it again, and around this process will go. The active site can lower an energy activation barrier by orienting substrates correctly, straining substrate bonds, providing a favorable microenvironment, and covalently bonding to the substrate. The rate of an enzyme-catalyzed reaction can be sped up by increasing substrate concentration. When all enzyme molecules have their active sites engaged, the enzyme is saturated. If the enzyme is saturated, the reaction rate can only be sped up by adding more enzyme. An enzyme's activity can be affected by general environmental factors such as temperature and pH, and chemicals that specifically influence the enzyme. Each enzyme has an optimal temperature in which it can function. Each enzyme has an optimal pH in which it can function, and optimal conditions favor the most active shape for the enzyme molecule. So here they're showing you a couple optimal temperatures for two enzymes and an optimal pH for two enzymes. So here the optimal temperature for a typical human enzyme, 37 degrees Celsius. Optimal temperature for an enzyme in a thermophilic heat-loving or a heat-loving bacteria might be 75 degrees. Optimal temperature for enzymes, pepsin in the stomach, might operate at a pH of 2, whereas trypsin, an enzyme that works in the intestines, may want to function at a pH of 8. So again, these are going to work in their appropriate environments. So cofactors are non-protein enzyme helpers. They may be inorganic, such as a metal in ionic form, or organic. An organic cofactor is called a coenzyme. And coenzymes include vitamins. Competitive and non-competitive inhibition. Competitive inhibitors bind to the active site of an enzyme, competing with the substrate. So the substrate can't get into there to do what its intent is. The competitive inhibitor binds to the site and prevents the other substrate from binding to it and changing its shape to make it something active. Non-competitive inhibitors bind to another part of the enzyme, causing the enzyme to change shape and making the active site less effective. Okay, some examples of inhibitors are toxins, poisons, pesticides, and antibiotics. So here, if you have a normal binding site, here's your enzyme, the substrate. Okay. Now, with competitive inhibition, this piece that would normally bind here can't get in because you have a competitive inhibitor blocking it from doing so. And non-competitive inhibition, you have inhibition because you have a non-competitive inhibitor binding to a different site on the enzyme, and thus it changes the shape such, notice the shape here and the shape here, changes the shape such that the substrate can no longer bind to it. So enzymes are proteins encoded by genes. Changes or mutations in genes lead to changes in amino acid composition of an enzyme, and altered amino acids, particularly at the active site, can result in novel enzyme activity or altered substrate specificity. Under environmental conditions where the new function is beneficial, natural selection would favor the mutated allele. For example, repeated mutation and selection on the B-galactosidase enzyme in E. coli resulted in a change of sugar substrate under lab conditions. So here you've got two changed amino acids who are found near the active site in this region. Here's your active site of interest, and there are two changed amino acids found on the surface over here, and two are found on the active site. Regulation of enzyme activity helps control metabolism. Chemical chaos would result if a cell's metabolism pathways were not tightly regulated. A cell does this by switching on or off the genes that encode specific enzymes or by regulating the activity of enzymes. Now, allosteric regulation may either inhibit or stimulate an enzyme's activity. Allosteric regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. Most allosterically regulated enzymes are made from polypeptide subunits, each with its own active site. The enzyme complex have active and inactive forms, and the binding of an activator stabilizes the active form of the enzyme. The binding of an inhibitor stabilizes the inactive form of the enzyme. So here you can see allosteric activators and inhibitors. You have an allosteric enzyme with 4 subunits. This is the active site, right? You've got 4 of them. You've got a regulatory site in between here. There's 4 of those. And by putting in an activator here into the regulatory site, you can stabilize it into its active form. However, these can go back and forth vertically and change, and this could be the non-functional version or the inactive form of it. And if you put an inhibitor in here into the regulatory site, it can actually stabilize it in the inactive form. So you can use an activator to stabilize it in the active form, and you can use an inhibitor to stabilize it in its inactive form. Cooperativity is a form of allosteric regulation that can amplify enzyme activity. One substrate molecule primes an enzyme to act on additional substrate molecules more readily. Cooperativity is allosteric because binding by a substrate to one active site affects catalysis in a different active site. So here's cooperativity, another type of allosteric activation. So here's your inactive form. You can see the substrate here is going to bind into a binding site and stabilize it now in its active form. So they're working together. In feedback inhibition, I think this goes back to chapter 1 or 2, the end product of a metabolic pathway shuts down the pathway. Feedback inhibition prevents a cell from wasting chemical resources by synthesizing more product than is needed. So here, if you, let's see, where should we start here? Okay, so you have the initial substrate here, threonine, which it's in its active, threonine is going to bind to the active into the active site of enzyme 1, which is threonine deaminase, which will go through a variety of different intermediates and different enzymes along the way, and then you finally get to this end product called isoleucine. Now, isoleucine will bind to the allosteric site on the threonine deaminase, which will make the active site no longer available and shut the pathway down. And then isoleucine will be used up by the cell eventually, and then the active site will be made available again to run through this process. Okay, so it's a negative feedback loop. The inputs will shut down the outputs eventually. Structures within the cell help bring order to metabolic pathways. Some enzymes act as structural components of membranes. In eukaryotic cells, some enzymes reside in specific organelles. For example, enzymes for cellular respiration are located in mitochondria. And here you can see the matrix contains enzymes in solution that are involved in the second stage of cellular respiration, which we're going to talk about here in the next chapter extensively. You've got mitochondria, so this is a mitochondria in here, and enzymes for the third stage of cellular respiration are embedded into the inner membrane here. So you're going to have enzymes in different locations that are going to be involved in cellular respiration, which we'll see in the next chapter. Thank you.